Hello, everyone. A very warm good morning, good afternoon, or good evening in whichever part of the world you are. I would like to welcome you all to the extensive 3 days workshop on NGS single cell data analysis and scRNAseq project support. This workshop is designed to highlight the efficient and effective data analysis using T-bioinfo workflows to analyze single cell RNA-seq data. So, twice a year, our community meets to review innovation in bioinformatics education and provide students with an outlook on careers and research trajectories rooted in computational biology. The Omics Research Symposium is an event aimed at bringing together this network of experts, educators, and students from all around the world to learn from each other, interact, and discuss advances in this field. And this is the time period we hold various workshops to prep up students, researchers, and faculty. So in today's workshop, we will learn about an overview of the current scRNA-seq technologies, basic overview of pipelines for processing raw reads into expression values, quality control of single-cell RNA-seq data, dimensionality reduction, and clustering techniques. We'll have practical sessions that will use case study approach using curated project datasets to learn about generating a table of expression from raw FASTQ files and perform subsequent analysis of this table of gene and isoform expression. We will cover standard statistical methods of analysis like differential gene expression as well as advanced data mining and machine learning for large-scale datasets. So before we begin with today's workshop, let me introduce myself and Pine Biotech for those who are joining us for the very first time. My name is Sunalika Ray and I'm the Omixlogic community manager. I'm responsible for daily interaction with thousands of our users that are a part of our bioinformatics community that is managed by Pine Biotech. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. Omixlogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. And over the past several years, we have had the privilege of working with a number of organizations developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. The training has been completed by over 15,000 participants from 162 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The training follows a project-based learning approach using research-grade tools to analyze data from top peer-reviewed journals. It has been completed in 6 different specialization tracks, which include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Now, to introduce the founders and the team members of the company, Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of inflammation. And one of his major projects is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a hard work by some of the people mentioned here on the right. Julia Pana, who is an expert bioinformatician, Avi Titovsky, who leads the development of the T-bioinfo platform, Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in top peer-reviewed journals. He has been developing collaborations with universities as a mentor and project lead for Omixlogic training and research fellowship. Dr. Raghavendran is an accomplished computational biologist with an experience in large-scale analysis of multiomics data and data science methods in precision oncology and protein DNA structural bioinformatics. So now let's gear up for today's session. First of all, you all need to sign up on the Omixlogic Learn portal that will give you access to the Omixlogic courses, which includes resources on bioinformatics, precision medicine, multiomics, including genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, and example projects of different fields, which you could learn from and replicate for your research work. Throughout the workshop, you will be given the directions to access the T-Bioinfo platform for bioinformatics processing and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. So now, before I hand over the stage to the mentors to get started with the session, let's just quickly sign up on the Omixlogic Learn portal so that we can follow along the resources that are shared by the mentors and also practice along. So for those who are joining for the very first time, I'll just paste the link in the chat box. Thank you, Ojasvi. Now let me give a quick demo of how you can create an account on the portal real quick and get started with the training with the access to the resources. So by clicking on the link that Ojasvi has shared in the chat box, you will be able to look at this page. You need to click on the login button that will redirect you to create your account. Now, either you can create an account for yourself by putting in the details required or simply use any of your social media accounts to sign up. I'll be doing it with my Google account. For those who have already created their accounts, please let me know in the chat box by sending a 1 or yes maybe so that I know that you have an account already. And others can take their time to create an account for yourself so that you can follow the resources that the mentors talk about today in the session. I'll be waiting for the responses in the chat box. Thank you, Darshini, for your response. For those who have already created their account, let's just quickly look at how you have to have a complete profile that will help you with your certification process and also help us monitor your progress. So when you click on your mail address that is given in the top menu bar, you will be redirected to a profile section. You need to make sure that you have an updated profile picture. Your name should be in title case because this is the same format that will reflect in your certificate. Please make sure that your name is not in capitals or small letters. Connect your social media accounts and also add a brief bio about yourself that could include your educational background or your research interests maybe. That will help us and the mentors to learn more about you. And with every coursework that you take on the portal, you will be able to look at your progress here in your profile section. And as a part of the workshop, you should be able to look at the badge of the workshop in your profile, which will give you access to the recordings and also the resources for 1 month. Now let's begin with today's session. Now I would like to pass on the stage to Mr. Ilya Brodsky, CEO Pine Biotech, who will talk about single-cell transcriptomic data analysis research support. Over to you, Ilya.
Thank you, Zunanika, and welcome everyone to this session. I will start off by giving you a brief introduction to single-cell seq, just one of the types of data that you can analyze at the single-cell level. And I'll focus on both some of the analytical challenges and the research questions that are being now discovered. I'll introduce some data sets. I'll talk in detail about some conceptual things that you have to know when you're trying to analyze data. And I'll introduce you to the resources that you can benefit from just trying to understand how methods of analysis actually fit in to the full overview of how this data should be analyzed and what are challenges associated with that. So as Sonalika already mentioned, we have a portal called Omixlogic that is focused on the logic of analysis behind many of these trending omics data types. And the reason we have this portal is to help researchers that come from a more of a biological background to appreciate some of the methods in computational biology and find data sets that could help them advance their research ideas. So we hope that as a student, as a researcher, you will be able to learn the basic concepts and quickly try to practically apply these analysis methods to a data set that you have selected. So that's what we're going to do today. We're going to look at the analysis part, and we're going to look at the data side as well. And the second reason is because we believe that the ideas in computational biology are becoming an integral part of any research. And so the data part of it, the data science part of it is critical. But to get started, you don't have to be an engineer. You have to be proficient in coding. You don't have to know everything there is to know about statistics, but you do have to develop an intuition about these different approaches. And the second audience that I'm going to talk to are engineers, people that understand the technical part really well and would like to understand how to apply what they know about the data analysis to specifically biomedical research data. And I think single cell data really represents one of the major trends that we are experiencing right now with explosion of computational challenges, methods, and approaches that any engineer can participate and bring a lot of effective solutions to. So really an exciting field to be a part of. So I'll start off by introducing the coursework that we already have. So those of you that already know bulk RNA-seq will benefit from advancing your kind of practical hands-on experience with single cell data. And the reason is that it builds on a lot of the concepts that you should be familiar with from basic transcriptomic data analysis, but you really have to read both the differences in the data and some of the methodology that is going to be used to analyze, identify patterns, and then be able to interpret biological patterns. What could you actually find in this kind of data, right? That's the question that we're going to talk about today. So in the bulk RNA-seq, as you remember, the major objective is to process reads that from a tissue or a collection of cells, so let's say cell culture or tissue, and then try to understand, typically at the difference between condition and control, what are some amplified pathways or gene sets. So that's the standard approach in RNA-seq, right? You kind of have to create a matrix of gene expression, levels, and then you can compare and contrast between different conditions. Now, in cell, what happens is that the resolution of your data becomes much, much more specific. And so I like this analogy that I heard, that instead of measuring temperature and averaging it across the whole hospital, right, where I can say I have dead people in the hospital, in the morgue, I have people with fever, very high fever, in maybe the infectious disease department, and I have average temperature maybe in other places as well. So if I measure the average temperature across all of these different conditions, I will get a normal temperature. But at single cell level, I am going to start to distinguish between the departments, and I can say, here's my acute patient department, here's the dead, and here are the normal people, right? So I can start to distinguish. And that is really one of the benefits of having such resolution. But how do we achieve that? How do we actually produce data that can help us see some of these differences? Well, in 2019, I believe this paper came out that kind of did a review of many different methods of single cell data preparation. And you will see here how first, these were very manual methods. So methods that people had to, take individual kind of several cells and try to separate them and so forth, and all the way to more and more automated methods. And so today, when we look at type of data that you are typically going to encounter, it comes from a specific machine that is designed to optimize, automate, and increase the number of cells and the accuracy of barcoding. But here you can see that, throughout from 2009, which the first paper that was published on Singlecell is in the middle of 2009, all the way to 2017, you can see an exponential growth of these single cell approaches. And so in 2017, actually, when this paper was published, they compiled all of these protocols that went from, very manual again to more and more automated. And you can appreciate now that whenever you take a data set that is single cell RNA-seq data, it actually could mean many different things. So to really appreciate the different approaches that have been more or less established over the past decade or so, maybe a couple of decades now, let's take a look at some of these basic ideas and technologies and try to understand how do we capture this precision and this dimension of single cell. So one of the first ideas is to differentiate between cells, right? If we are going to study something at the single cell level, we need to understand how do we differentiate between many thousands of cells that we potentially have to sequence. And to do that, one of the ideas is to give them a barcode. So if I could generate a barcode for a type of cells, right? I'm not generating a barcode for every individual cell. I'm just assigning them to maybe different tissues or different batches or something like that. So here I have barcodes. For example, I want to have a bunch of cells, and I have a tumor, and I have a microenvironment, right? And I want to be able to differentiate between them. I can give them a barcode, and those barcodes have to be encapsulated with the cell, right? And so if I kind of take a droplet, let's say an oil, droplet of oil, I can put a single cell into that droplet and a barcode bead. And that barcode bead looks like this. It actually is a bead that has these connected DNA strings attached to it. And once I prepare my data like this and I keep them all inside the same bead, now I can sequence individual cells. So this is the basic idea, right? But now let's take a look at three different methods I think I have here. So the first method is called Microwell. So the benefit of Microwell is that you actually end up with a plate, very similar to what you would do with microarrays, right? You have a plate, it has these small wells on the plate, and you're hoping to put in an individual cell within that microwell. Now, this is a process that could be optimized, and you can see in this case, this is that oil droplet, and here is that little cell, right? So that little cell right here, magnified over here, you can include additional phenotypic information. So essentially, in this case, you can make different color, fluorescence, and you can potentially identify an additional aspect just to give you a control, right? But in many cases, the limitation of this technology is that you only have a certain size, right? You can have limitless well plates, so you have a limited size. And so the number of cells is going to be quite small, right? So if we're talking about thousands, maybe hundreds of thousands of cells, you're going to be limited by the number of wells you have and the number of cells that you're going to successfully place inside each individual well. The second technique is called microfluidics RNA-seq. And so microfluidics really tries to separate everything, again, in a plate. So you have a plate, and that plate is where you put your cells inside this area right here. And then they start flowing over here. And as they flow through, you can see that you put in the cells, they start going in, and they get trapped. And so these traps right here is where you will end up having those individual cells. And those cells, again, are going to be, well, these are going to be cells that you would hope to then sequence at the end of the process, because then you can separate them. Okay, so this is that second option, right, when we have microfluidics. The final one, and probably one of the most successful ones, is the droplet single cell RNA-seq data preparation. And the idea here is that you have a continuous flow of drops. And here you can see that the cell and the hydrogen are incorporated into here. And as they flow, you can monitor them visually, and you can separate the ones that do have a cell in them or don't have a cell in them. So you have greater control over this. And you have here, you can see that it kind of encapsulates them into this oil bead, and then you can flow through and add the barcoding. So this is really a way to scale up and automate this whole process. And so the idea is that you encapsulate each individual cell inside a nanoliter droplet together with the bead. Then you can isolate the bead with the enzymes that are required to construct the library. And each bead will contain that unique barcode that, because this whole process is going to be automated, right? And at the end, you have this ability to separate your reads into individual cells, right? And this is the technology that is going to be used most often today when you encounter single cell data. For example, 10x Genomics is going to be utilizing something like this. Okay, so the second part that we're going to talk about is the basic processing of such data. So let's take a look at the basic processing of such data and try to understand what are some of the critical steps that are required for us to extract a meaningful matrix of cell-specific expression of genes. And then we'll think about what are some of the challenges associated with this process. So I would separate this process into three major steps. There's a process of when you align the reads to the reference genome. Basically, that's the idea. Number two, you then normalize, because you have so many different cells, so you want to normalize the data. And once you normalize the data, you then want to assign them into particular clusters, right? So the idea of clustering, we'll return to that later, how to perform clustering in a meaningful way. That is the overall kind of review. What are some of the issues that we're trying to address at each one of these stages? So let's first of all talk about this first stage, the stage of preparing the reads, aligning them, and making sure that we're only taking reads that are aligned correctly to the reference. So as in any process of alignment, we will have FASTQ files, right? So in this case, we already have FASTQ files associated with each individual cell. We then have to align these reads to the reference genome and generate a BAM file, right? What is a BAM file? A BAM file is the same FASTQ, but it now tells us the beginning and the end of each read from the perspective of the genome. So you have the starting position and the ending position on the reference genome. Now, instead of using here actual individual reads, we now switch to UMIs, right? And UMIs are adapters for each mRNA. So UMI helps us determine how many mRNA pieces are present in each cell. So it's like an alternative to the read. So instead of counting reads, we count UMIs in each cell. And that gives us a better sense of what level or what abundance of mRNA we actually observe in more of a biological sense than just counting those reads, right? And so here you can see that each read contains the barcode, the UMI, and so we count these UMIs. Now, we have thousands and thousands of cells, right? So then the process to count these UMIs has to do with comparing cells to each other, right? And the problem typically is that we don't have enough RNA, mRNA material in each cell. And so here we run into some challenges, right? And the challenges are going to be to assign those UMIs correctly, and then to have as much data as possible so that we can actually take the high quality data to make biological interpretations, right? Because if I, for example, want to identify a cell type by a marker gene, but that gene doesn't have enough mRNA, it's hard for me to distinguish between low expression and a missing data point, right? And so that is one of the challenges that we're going to run into here at this stage of counting. And so an important part here is to really look only for cells that have a sufficient number of umis that come from gene coding mRNA. So typically here we're going to be looking at just gene expression, not transcript level expression. So an important part here is going to be to filter out only cells that have a sufficient number of genes that I can assign an expression level to them based on the overall number of my gene expression. Can everyone see my screen? I see here on the chat if I can share the desktop. So I think everybody can see my screen, correct? Okay, great. All right, so, okay, so let's take a look again at the overall process of getting raw files into a pipeline and getting a matrix at the end. And let's think about what we can do with that matrix after this part. So first of all, we need to identify what essentially are we trying to get at. And we're trying to get a table of expression. So instead of samples, we're going to have genes. So typically in a table of expression, you have genes and samples. Here we're going to have a transpose table, and we're going to have cells and genes. And once we have only for cells that we have sufficient data for those specific cells, once we have a sufficient matrix, we want to check for artifacts, right? And those artifacts are going to, again, help us prepare a better table of expression. And that table of expression will be, as many cells are going to be filled with numbers and not zeros. And that's a major challenge dependent on these factors of data preparation. Once we have that, just like we do in bulk RNA-seq, we're going to normalize the data because we know that the difference between mRNA levels are not always coming from biological differences. They could be coming from these technical variability issues. And finally, we want to then understand difference between groups of cells. So let's now take a look at how do we go to this downstream analysis. Basically will fall into these three major challenges of analysis: clustering, annotation, and analysis of a trajectory. And we'll talk about some use cases where these approaches are going to be most useful. So first of all, clustering. Briefly, I'll give an overview, and then we'll pause to take some questions. So for clustering, one of the major challenges is going to be how do I, what do I call similarity, right? These are going to be heterogeneous samples. And so I have a lot of variation just at the level of expression. I have a lot of variation inside specific genes at the level of expression. And now when I try to find differences or similarities, before I can do that, because in such a, I have, again, tens of thousands of cells here, right? So it's very difficult to work with such complex data. Before I do that, I want to reduce the dimensionality. And we'll talk about two methods, t-SNE and UMAP, to reduce the dimensionality. And then once I do that, I want to perform a specific type of clustering. So depending on the method that I choose for dimensionality reduction, depending on how many cells I have and how complex the data is, I can choose different approaches for clustering. Once I determine that, for example, this is a cluster and this is a cluster, I need to annotate them, right? And annotation means to assign a cell type, typically. So when I want to assign a cell type, I want to select marker genes that are coming from some database, from some publication. And this is a major challenge in single-cell RNA-seq, is appropriately assigning a cluster that is based on a statistical analysis of my data set to a particular of tissue, cell, or condition in some cases, right? A cell in a condition. And the final kind of analysis that you can perform in some cases is to try to determine exactly where in a trajectory of development a particular cell group is now in my sample, right? So why is this an important thing, and why do we not do this in bulk RNA-seq? Cells are not static. Cells are dynamic. And I have, let's say, a group of cells, let's say in a tumor, right? Cells are proliferating actively, and they are found in different stages of proliferation. So even though they are cellular cells, for example, they could be in different stages of differentiation. And I want to oftentimes find a process that I could find individual cells that have multiple representations. Now, this is going to be very appropriate for a time series data set where I can take, for example, some progenitor cells and I can see their differentiation over time. But sometimes it's also interesting to see a process in a complex population of cells. And I know that these are all cells from the same tissue, but I know that they're differentiating at different rates. And so I'll find some that are in the beginning and kind of toward later stages. Okay, so let's pause here. And let me ask you if you have any questions before we jump into some of these data sets that we can leverage to try to understand the differences between different types of cells. All right, so we have a question here for human cells for annotation, cell type, or classification. Is that where we may utilize the human cell atlas? Yes, and we're going to look now at some of these different compositions or these atlases of cells and try to see what are they trying to achieve and how we can use them. So, here you need to think of kind of several different challenges. First of all, as we know from collections of 100,000 human genomes, 1,000 human genomes, HapMap, TCGA, all of these collections, what did they reveal to us? Every single one of these projects was designed to map out the heterogeneity or the variability of different populations. And we said that, you know what, let's solve cancer. We'll collect thousands of samples of human cancer tissue, and we'll understand what does human cancer look like. What we discovered is that we don't have enough representation, right? And so the idea that we can comprehensively map out what's happening in biology is almost impossible to imagine, because by the nature of biology is continuous evolution and difference and uniqueness, right? And so it's very difficult to map out comprehensively everything that we would like to identify. So that's the major challenge. If you take the single cell level, the diversity increases many folds. And so that is where you're going to find that challenge as well. But yes, the idea is to try to map out as much as possible, and either from publications where people biologically validated these markers, we can find marker genes for different cells or different cell types. In many cases, we try to approximate. Okay, the clustering step is done based on which criteria? Great, so clustering in general looks at similarity between expression profiles, but in this case, since we have many, many genes, many, many cells, we first reduce the dimensionality. And we say, let's reduce the dimensionality, and in that hyperplane that we have created, let us find some distance metric, and we'll look at several different methods today, and we'll look at what goes into those methods to then quantify something as belonging to a particular cluster. Okay, Rajan is asking, how do we annotate any novel cell population if we don't have any database available? I guess some de novo algorithm pipeline to identify novel cell. Yeah, so a great point, right? So in many cases, right, because we have limited annotation, we cannot assign a whole group of cells, right, many, many cells to any particular type that is known. I mean, the idea is that actually that is the objective of single cell RNA-seq, is to discover new things, not just confirm what we think we know. And so here it's going to be very important to have clustering that leads to discovery of markers, right? And then we can biologically try to validate. And in fact, we'll show today, we'll look at today at some of these repositories, they also identify novel types of cells as well. And that's an important discovery to make as well. Okay, we have another question. Okay, liquid biopsy-based single-cell sequencing for circulating tumor cells for cervical cancer. For that, I'm looking for single-cell sequencing facility, if anyone knows. Okay, so yeah, there are many single-cell facilities. So Tanzil, either someone here in the group, or we will be able to connect you with someone, hopefully in India, I think. Okay, so maybe Sonalika, you could point them to the right direction. Okay, so if we don't have any more questions, let's take a look at some of these references, right? Where do we get these actual gene sets and markers, and what is found in there, and what are these efforts? So first of all, let's look at one of these projects. Oh, sorry. One of these projects is going to be a map of human tissue. So this is the publication. Let me just share. Let me get out of here real quick, and I'll just share the link to this publication for those of you that are currently working on this area. I think this is going to be a useful publication. Okay, so here we have a publication about this effort to try to make a single cell type transcriptomics map of human tissue. So you can see that this is from July 2021, so a very recent article. And you can see here the humancellatlas.org, right? And this is something that we can actually look at. Let's try to understand, because we have this capability to sequence individual cells, we would like to create a reference that is as comprehensive as possible of human tissues. And so this particular effort is trying to create an open access atlas that will allow researchers to explore the expression of human protein coding genes in 192 individual cell type clusters. And then an expression specificity classification was performed to determine the number of genes elevated in each cell type, allowing comparisons with bulk transcriptomic data, right? So there's a lot more data already that exists on bulk RNA-seq. And in this case, we would like to take these single cell data and increase the kind of specificity or the detail of this bulk RNA data. And so they took, you can see here, both imaging, bulk RNA-seq, and single cell data to try to dissect all these tissues into individual types of cells that then we could reference and understand specific genes that are elevated or downregulated. So let's actually take a look at this. This is the place that you would go to find the actual data. And you can read here a lot of the sources of the data that they used. And also you can see some of the examples of the data. And the examples of the data are possible to view and visualize. And so that's what we'll do today. What we'll do today is one of these collections, because all of these are going to be collections of data. And let's explore what does it actually look like. So if you go with me to this second link. Okay, so I'll go to this link. Yes, yeah, so Nihal, that's exactly correct. We're trying to build a reference. So let's try to understand what this is. This is, by the way, I believe this is, let's see. Okay, layout, new map. Okay, this is oligodendrocyte, right? You can actually select many different types of tissues, but let's just take a look at here specifically. What can we find? So when we think of types of cells, right, we typically say, okay, well, there's neuronal cells, there's oligodendrocytes, there's microglia, macrophages, etc., right? And you can see here what in reality we find, right? In reality, this type of analysis helps us increase the dimension. So for example, you can see oligo 1, 5, 2, 6, 4, 3. So there's 6 different types of oligo cells, right? And you can see here that, if you actually select, they mix a lot between themselves, right? These are not clusters that are, always very, very clearly defined. I mean, you can see that all of them are right here, but they're also found right here. So a lot of these types of references give us the strongest signal that we have. Not all of them are so clearly differentiated from each other, right? Some of them are, but not all of them. Okay, so here, for example, endothelial cells, microglia, right? But some of these other types, which are probably the majority of the cells, start to be more spread out and more complex, right? So the more we want to define these specific cells, we can group them into larger kind of collections of different subtypes, but also within each group, this larger group, we will find more and more specific trends and patterns. Okay, so this is healthy tissue. Okay, so remember, this is an attempt to map out all of the different cell types, different bulk RNA-seq data, imaging data, and you will always see the differences in morphology and kind of the different phenotypic part of that data as well. But then you want to go deeper and deeper and try to understand, well, what are the differences within this, big collection of cells that we call neurons, right? Obviously, there are many different types of neurons. Okay, so the next thing that we want to look at is look at some examples from disease. Okay, so here we saw these are healthy tissues. Now let's look at disease. So this is a collection of data that was taken from all of the possible data sets that are available on tumors. So in cancer, we would like to now understand how different types of cancers, which we know are primarily by tissue type, right? So when you have breast cancer and lung cancer, the differences between those tumors are going to be associated with the type of cancer, lung or breast. But now we would like to understand how these cancer cells differentiate within a particular subtype. Okay, so this is, again, something that you can find. I'll post the links here as well. This is a project by some Chinese university where they basically re-annotated, right? They collected a lot of meta-analysis, of data sets that came from GEO, from TCGA, etc., right? And they tried to understand, again, what is the relationship between what we know from bulk RNA-seq to single cell? So again, let's take a look here at one such example. So here we'll take lung tumor microenvironment, and we'll try to understand here, what can we learn about, a condition like cancer, which is a complex condition that we can actually try to interpret biologically. So what I'll do is I'll go to this link. Again, on your own free time, you can explore all kinds of other types of data that are available here. And you'll find that, a lot of these are just collections of data sets that others have published. Okay, so here you will find a typical analysis workflow, and we will, in our second session today, learn about the different steps of analysis and how to actually perform them. But in my session, I'm going to ignore some of these basic things, and I'm going to move on to kind of like, what do we learn from these collections? So again, you will find here we have some dimensionality reduction with t-SNE and UMAP. You can see that UMAP is typically going to be a little bit better, making the separation greater between the larger groups. But actually, if you compare the two, it's obvious that both are useful. For example, here I have a greater resolution of this mixed cluster. And here this mixed cluster is very condensed together, right? So if you scroll down, let's just take a look at this chart right here. And let's see, so again, this is the tumor microenvironment. What is the tumor microenvironment? The microenvironment is like the layer between the tumor that kind of created its own hyper growth, aggressive, this kind of like uncontrollable tumor that is hurting everything, and the normal tissue, right? In between, there's this area that we call the tumor microenvironment. Typically, it has a lot of immune cells, right? So when something like that happens, the natural reaction of the organism is to address it by putting there the offensive, right? And saying, let's first of all quarantine this little place, and then let's kill them. And so you see here T cells, you can see here macrophages and other kinds of cells, right? And specifically, you can see here, if you click right here, that there's about 3% of malignant cells, right? Why is this important? Well, this is important because a typical approach for treating cancer is reducing the tumor size through chemotherapy and then trying to cut out surgically whatever we can that makes that a potential for new growth, right? And so typically that does not include all of the microenvironment because it's difficult. The microenvironment is integrated in healthy tissue. It's difficult to separate it, but that is the problem. The problem is that you have 3% malignant cells that are inside infiltrating the tumor microenvironment. So if you think about cancer and the stages of cancer, it's all about this proliferation and kind of going beyond the boundary. So containing the tumor in a well-defined boundary is stage 1. And as it goes beyond that boundary, that's when your stages continue. And eventually metastasis is that they're way beyond, right, the actual environment. And so this is the problem right here, these malignant cells. Now, you can see here, this is just counts of cells. If you scroll down, this is now differential gene expression, right? So now we would like to ask, well, what are the differentially expressed genes in these different clusters? So these are the different clusters, and let us try to compare them and identify differences between the clusters. And here's what we have, right? So these are gene names. These are types, or these are the clusters that we have. So let's find here those malignant cells right here. You see, this is the malignant cells, right? So this is this cluster right here. And here we have a group of genes that are unique to this cluster, right? So we can find these specific genes and we can say, okay, whenever I look at lung cancer, let me look for these specific genes and I will be able to find some cells that are specific to this malignant type. So where do we find them here? Here, you can see I can find them malignant right here, malignant cells. So they are very different. And that is because if I could look at the tissue and say, here's the group of malignant cells, they're not going to be grouped together. They could be spread out, right? But I can now identify them. And that is very important. Okay, so this is an example, again, we have for cancer. And you can take a look at other examples as well. Now let's take a look at another major condition. Another major condition that has been, on the top of everybody's mind is SARS-CoV-2. So here's a collection again of all of the data sets that are available for COVID-19, and I'll paste a link to this here as well. Okay, here we can see how we are trying to map out the developed disease in different tissues, right? And you start seeing these UMaps that look like this, which is now a lot more difficult to interpret, right? Because there's so much overlap here. But essentially what we can do is we do a comparison like this. And in this comparison, we have, it's a single data set. So again, remember that there's a lot of variability in the data sets, but here is the basic approach. Let's take some COVID-19 patients, take some healthy patients, and let's try to count how many cells of different type do I have. And so what stands out to you in this comparison right here? What difference do you notice most between these different groups of samples? Who would like to take a look at that and suggest the differences that you observe in this chart? So again, okay, so Julia is saying endothelial lymphatic cells higher. How did she get to that idea, right? Let's take a look. So first of all, we're looking at colors. Inside each color box, we have number of cells, and the colors are actually annotated down here below. So here she's saying endothelial lymphatic cells. Okay, so this is this purplish color right here, is amplified in COVID and almost doesn't exist, right? Maybe right here, almost does not exist in healthy patients. You can also say the biggest difference that I observe is this orange color that is much less in COVID compared to healthy. So here, what is the orange? I go back over here, alveolar, right? So what kind of tissue is this? This is lung tissue. So what are we seeing? We're seeing that COVID-19 is killing off, right? Eliminating cells that are alveolar. You can see here expanding types of cells. So for example, red, right? So here I can see an expansion of red cells. What are these red cells? These are natural killer T cells, okay? So we kind of now can have a direct biological interpretation. Well, we know that COVID replicates in alveoli primarily, and then we see the natural killer T cells are coming and attacking them, right? And that is immediately clear from this particular study. And that's an exciting thing to see because it really confirms, what we kind of know intuitively from reading these publications. We see where it's coming from. And obviously, like Julian noticed already, a lot of these other types of cells have also very many differences as well, right? So now that gives us an insight into what additional questions could I ask, right? How could I think about this and expand on this, basic level that I already know and start asking more specific questions? Okay, so these are some collections, just examples. There's also a mouse cell atlas. So a lot of work is done on animal models, right? So there's a lot of information about mice, a lot of information about human healthy tissues, some disease conditions as well. You will find the developmental aspect is very interesting as well. Development of different types of tissues, development of disease, etc. Let's take a look now at a publication where we will see a connection between a phenotype and single cell level data. So just an example, but this will help us think about how people actually use this kind of information in a practical example. So this is a project, it's not very new. It's from 2017. And you have to remember that all of these technologies to generate data are expanding over time. And, 2020, we're 2022. So it's been now 5 years, and 5 years, single cell technologies have been advancing rapidly. But this is a great example of a kind of standard use case for single cell data earlier on that was, integrating the parameter of how an organ functions and how we can observe the differences at single cell level, which is an exciting and interesting way to think about this. So this publication is called Detecting Activated Cell Populations Using Single Cell RNAseq. And it's talking about neuronal circuits. What are neuronal circuits? Neuronal circuits are essentially connections between neurons in different parts of the brain, right? So our brain is a pretty big organ right here in the head that has many different components, and they have these very specific functions. But our brain is not like a hand that works separately and then a leg that does completely something else. It kind of works in an integrated way. And so here, when we want to study a behavior, they wanted to understand how neuronal circuits, their mechanisms and pathways are involved in behavioral response and are really critical for our understanding of anxiety-related behaviors in rodents. So here they took a mouse or mice and try to understand how that happens. So to do that, they actually took single cell data. They sequenced different regions of the brain. And because the cells are spread out and they're very different, they wanted to understand specifically what is the functional property of that cell. And they wanted to associate that with behavior. So first of all, let's look at these circuits. So this is what these circuits actually look like. You can see here different parts of the brain. They kind of interact with each other. There are many different things to kind of understand here. But in this particular project, they were trying to understand how a stimulus actually could be observed at this basic biological level, right? How do we kind of take this complex behavior and map it out at this particular level? So they took 3 different types of data. Morphology is illustrated here. So morphology is how these different cells look, right? What do they look like under a microscope, right? Physiology, and physiology here was measured by electric signals that could be measured. So it's the property of electric signal being passed through this complex structure, right? And you can kind of see these patterns. And the final one is to connect it with a molecular signature. So what genes actually function, what are amplified, downregulated, and how can we connect those 3 things together, right? A very interesting project to really, show that this functionality is regulated by particular genes, particular proteins that are being encoded in these genes. So this is what they did. They did a single cell analysis of neurons. They did fluorescence in situ hybridization, or FISH, and then they also did patch-seq. And so as a result, you can see again a familiar figure, right? So one of the major challenges is, let us collect all of the cells and let us try to differentiate between them by type, right? And assigning those types is a major challenge because we need to have specific markers. And then we would like to understand differences between these markers, right? How do we connect the differences between these markers with the actual phenotype? And so let me actually share here really quickly the link to this data, or to this publication. So this is the link. Okay. And to really appreciate this data set, I think it's useful to understand that integration of multiple types of data is ultimately one of the challenges and one of the opportunities for these kinds of projects. So ultimately, they did identify, as we saw, a specific pattern, right? Specific genes that are amplified, and they were able to, just like we saw here, they were able to say, well, this is how we can measure this at the molecular level. This is how we can measure this at the phenotypic level. This is how we can measure this at the physiological level and associate this with behavior, right? So an exciting finding. So this is just an example. So again, what did I show so far? I showed 3 different examples. One, a collection of data to map out normal variation in different types of tissues and cells in human and mouse. Number 2, we looked at examples of disease-specific maps or atlases, right? Cancer, COVID, and there are many others. And number 3, we looked at a particular example of a study where they try to use this type of resolution at the single cell level with asking a practical biological and clinical, in a sense, or psychological maybe question, anxiety and behavior. So are there any questions? Okay, so if there are no questions, let us take a look at, as you saw, in many of these examples, we find these pictures, right? This is a very standard picture. We kind of look at, okay, t-SNE, UMAP, right? We kind of tend to look at, this is the picture that defines single cell, right? We all kind of see single cell and we associate it with these images. So in fact, it would seem like you just put the data in, analyze with this method, and you get a result. But the reality is much more complex. So here we'll take a look. First of all, let's take a look at this publication that is called "The of Using t-SNE for Single Cell Transcriptomics." Okay, so I'm just going to go. I shared the link. Please take a look at this link in your chat box. Okay, so it turns out that these methods are not straightforward, right? You can't just, take this method, run it, and get the result, and everybody's going to get the same result. This is something that requires an understanding of the methodology and the limitations of the data as well. Okay, so please let me know once you've reached this paper. I think it's going to be very useful. Okay, so here we have an example of the same data set. It's a static data set that is analyzed by different methods. Okay, I think I can make this bigger. Okay, there we go. Maybe not. Okay, there we go. Okay, look at these. So your choice of methods makes a difference. This is MDS, or multidimensional scaling, PCA, principal component analysis. This is the default t-SNE, okay? This is when we change t-SNE perplexity, and this is then different approaches to PCA, right? So we can change parameters. These are called hyperparameters. We can tune them, and the results will change in terms of separation. So let's first of all take a look at this example. So this example, what is multidimensional scaling? Multidimensional scaling basically assumes multidimensionality. We can use something like NMDS, non-metric dimensionality scaling, or metric dimensionality scaling. But the basic idea is that we have counts of these UMIs, and we want to identify distances in either a metric, linear, or a non-metric, non-linear space. And here we can see that the differences are going to be dramatic. So typically when you do a dimensionality reduction approach, you then want to assign groups, and that is done through clustering, right? Somebody was asking, well, how is clustering performed? Well, typically, let's take PCA now. Let's take a look at this example, because here we really see some outliers primarily, right? And not very good clustering. Here in PCA, we see, again, this is synthetic data, we see very clear separation between major groups. And then we have an assignment of clusters. And it basically tells us that there are 3 clusters, and these are homogeneous clusters, right? So PCA works by linear dimensionality reduction and linear Euclidean distance. So this is assignment of clusters. If we take the same data set and we analyze it with t-SNE, we end up with many more clusters, right? So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 clusters instead of 3, right? So what do we see here? We see that the method that we choose will lead to different results. Now, if I'm a biologist, I will say, okay, my method of analysis identified 3 different groups. My question will be, well, what are these groups? How do I explain the differences between these groups? So I will measure differential expression between these two, and these. But here, I now start thinking, okay, well, this is many more differences. Now I have to perform many more different analyses and explore the differences. So really, when I try to go from the standard to interpretable results, I have to make decisions about parameters. And I have to make these decisions about parameters by understanding the parameters, not blindly, not because this is the standard setting, perplexity 30, random initialization, n equals 200, right? That's the standard parameter. Here I have a change in parameters, and you can see how that affects what I will get. Now, another thing to remember is that t-SNE works with already reduced data. And that's where this PCA part comes in as well. I feed my t-SNE with already reduced data. So typically, a standard approach would be a reduced dimensionality from all dimensions to 50. So I'll have a PCA top 50 principal components, and then I run t-SNE, right? And that also plays a role in what my outcome will be. So an alternative to t-SNE that has recently become popular is something that's called UMAP. Now, I'm sorry, I'm going to run over time just a little bit. Okay, excellent. Yes, so let's take a look at how do we understand these parameters, right? That's your question, basically. So let's look at this interactive thing that will help us understand some of these parameters. So this is, I pasted a link in the chat. And this is just an example. Okay, so again, this is for t-SNE. t-SNE typically takes in the data from PCA already reduced. Typically, number of PCs is going to be your parameter. Okay, so here what you can do is you can change the parameters and then you can run on a particular data set. Okay, so let's do the run just with the basic parameters right now. And let's see what happens. Okay, so this is the result. This is t-SNE, and you can move this around, by the way. Okay, so this is t-SNE, and this is the 30 top principal components. Perplexity, which is the non-metric scale that we use in, right? The distances that you see here are in perplexity. The learning rate and the maximum iterations. Because t-SNE can run forever, we need to limit it at some point, and so we limit it at 500. Okay, did everyone run this first example? Did everyone run this first example with the basic parameters? Okay, after we run these basic parameters, obviously we want to, okay, great. So let's try to change. So again, like I said, a typical change would be we would increase the number of PCs to 50. So let's increase this to 50. Okay, and as we saw in the previous example, their perplexity was 100. So let's change this to 100. Okay, so let's take a picture of this. That way we will remember what this looks like. Okay, so just change those two parameters and let's click on run. Here, remember that you also have this option. We're not going to touch this now, but let's just click on run. Again, I'm going to pull up the original picture. Okay, so this is what we did with 30 and perplexity of, I think, also 30. Okay, so this is what we got now. Do you think this has improved, or has this been, or did it become worse? What do you think? Okay, worse. So Julia is saying it is worse. Okay, why do we think it is worse? What gives you the impression that this is worse? Julia. Harder to distinguish cluster. Okay, so what Julia is saying, okay, Julia is saying that visually, right, Julia? You're saying visually, I can see here the distance, right? The distance between the clusters. It's greater over here, and it's less over here. This is more kind of together. And that is why I am saying that this is a worse choice, right? This is a better choice, and this is a worse choice. Now, surprisingly, there's actually a publication on this that people kind of looked at how we tend to interpret some of these analyses, and people determined that actually that's true. Most people visually look at a more appealing picture and they come to the conclusion that this is better. Now, in reality, you don't know that this is better, right? What do you need to do to make your decision what is better, right? How would you, choose that appropriately? Well, there's actually an art to this, right? So it does depend, right? You're correct, Nicholas. So it depends on what? Depends on what the data is, right? What is this data? So the data, how do we work in biological experiments? The data always has controls, right? And so the first thing that I would like to know is whether my controls are grouped together, right? Now, in many clinical settings, I'm not going to have exact controls, but I'm going to have an idea of these controls, right? And so it's a lot more difficult than you would imagine. It looks like you look at these publications and they all have these nice t-SNE pictures, and they kind of see, this is a cluster here, and this is a cluster here. Everything seems to be, so easy and perfect. In reality, it is not. You need to have controls. You need to understand your data. In many cases, they're artifacts, and learning about the methods themselves and playing around with these kinds of parameters to understand, again, this is a specific data set, right? This is not just synthetic data anymore. We are now going to discover how these parameters work, and then you can apply them in different situations differently. Okay, and that is something that I want to be the outcome of today's session, is that you understand that this is a promising technology that is developing rapidly, and we need to understand the methods of analysis in detail to be able to apply them correctly. Does the size of the data affect this choice as well? Exactly, yes. And the size of the data, right? What are we doing right here with reducing the number of PCs or choosing the number of PCs? We're changing dimensionality of our data. So we're reducing data. Okay, so if I increase it even more and I run it, let's see what happens. This is what happens, right? So the more data, the less clear it becomes, right? So the intuitive thing would be to say that if my data is more simple, it is going to be easier to separate, but I'm reducing data, right? I'm reducing the amount of data and reducing the amount of information. And so it seems like if I reduce the amount of information, it's better for me. But is that the truth? Is that always the case? Not always, right? In many cases, it's the opposite, right? The more data I include, the better results are. So this is something to think about. Let's just quickly, I know that we're running over time, but I'm going to quickly go through another example. So in many cases, you will find another method. Another method is called UMAP, not TSNE. Okay, and this is going to be, again, something to play with in your own free time. Sorry, that was just to one person. Let's go to everyone in the meeting. Okay, so this is another example here to understand UMAP. Now, UMAP is similar to TSNE, but TSNE runs into these limitations of large and complex data sets, and we have to reduce it. And so we are kind of making the choice. UMAP does not have this kind of a limitation. I'm going to make this just a little bit bigger. Okay, and here, the idea is similar. You can essentially play around with a number of parameters using these synthetic data sets. So you can kind of see what parameters achieve what kind of result, right? And you can really try to understand intuitively, again, what are the specifics of these parameters and how to try to choose them. And here now you can also compare between TSNE and UMAP, and I think this is a useful illustration to kind of see how this works in a data set that is a lot intuitive than the actual biological data sets. So before you jump into the biological data sets, play around with this, try to understand what these parameters actually mean. You have a lot of many examples here, and you will see. And in the next session, by the way, we'll go into more technical details so you understand the math and kind of the coding principles behind some of these things. But this is something that will help you understand, how your data kind of theoretically should look like, and what does it become in the end. Okay, so Aditayo is asking which parameters are available on the TBio server. So great question. We will get to that in just a few more minutes, okay? Okay, so again, your understanding of methods, your understanding of the parameters of these methods, your experience working with a lot of examples is what builds your confidence when you actually come to a real practical challenge that you might see in your own data set. Okay, yes, more questions. What are some of the parameters that we can extract from TSNE or UMAP separately? From what I understand, it's better to run them both. Exactly, yes, I would agree with that assessment. You want to run them both. You want to compare and contrast multiple methods and then select something that fits in with your controls. Maybe go through several steps of transformation. And that is really why it is useful to have easy to run as opposed to being, stuck kind of dealing with the complexity of running everything independently. which we'll do today together. But, I think still it's beneficial to try and understand and then be able to compare and contrast. Exactly right. Okay, so we talked here about t-SNE and UMAP, these traditional figures that, we always expect to find in single cell data. And we understand that parameters make a big difference, oftentimes counterintuitively, right? Counterintuitively means that the simpler the data, the clearer the separation. The more complex the data, the more challenging it is to find the association between the data that we expect and the data that we get in the analysis results. But that doesn't make it wrong, right? That does not make it wrong. And we should kind of base our assumptions not on what's intuitive, like visually this looks nicer. Instead, we should be looking for, what do we want to understand through this analysis as we try to get to some biological discovery or insight. Okay, so what are some standard tools that you will be running across when you are using, your single cell RNA-seq data? What are some typical tools that you're going to be using? Well, typically, as I mentioned, a lot of the data is going to be coming from 10x Genomics. 10x Genomics have their own suite of tools, including Cell Ranger and this loop browser, that allow you to explore, analyze and explore your single cell RNA-seq data and make sense of them. And so this is an example, a screenshot. You can see it's 10x Genomics. Essentially, the approach is going to be similar, right? Let's reduce the dimensionality and let us look at either all of your data or sample-specific data. So again, remember that in each sample, you have thousands of cells. And let's try to categorize them by clustering. Now, the attempt to reduce the complexity of the analysis to a more user-friendly interface is only as useful as the logic behind your analysis. Your logic has to be solid, and then you can use effectively. This particular interface is useful, but it can be misleading when you have to adjust for facts, right? This is going to jump directly from here's your raw data, here are your UMI counts, here's your visual like t-SNE or UMAP. What you're missing is the ability to address the technical issues before you get to this step, okay? Now, to really address the complexity of these types of data sets, typically people use two major packages. These major packages include curated, which is a package that has been evolving over the past several years and now is in version 4. And that just gives you a sense of how fast this field evolves. So if you remember, major algorithms for mapping, for example, like bowtie, we only have bowtie 1 and bowtie 2, right? And that's it. Or, you have top hat, you have top hat 1, top hat 2, and then high sat 2, right? And this is over the span of decades. This, in a few years, you already have number 4. Why is that? Well, because along the way, people have discovered that there are these issues, these underlying artifacts that we have to be able to discover and address. New techniques allow us to select genes that kind of make our analysis more reproducible and more biologically interpretable. And so a lot of these kinds of changes are going to be included here, but this is going to be available not only just for RNA-seq, but also for other type of single cell data. Okay, so curated is a major package, and we'll talk about this in a little bit. And another package is called Microwell, right? So in the original attempt was to build a specific method of analysis with Microwell to just look at pathways of differentiation and cell differentiation, these trajectories. But today it evolved into a whole suite of tools, and these tools include trajectories, clustering, and differential expression. And so these are all important aspects of single cell analysis. So what we will try to look at today is the implementation of these different methods on the T-Bioinfo server, where you can look at the whole kind of step-by-step path. We're not going to run an actual analysis. We're just going to look at this pipeline so that you understand that it exists. And later on, when you learn all of these different methods, you will essentially learn how to reproduce these kinds of visuals. And then what we'll do after that is we will go into a Colab notebook that you will see the code behind some of these methods, okay? So what is the T-bioinfo platform? The T-bioinfo platform, for those of you that are joining us for the first time, is essentially a big data analysis platform that allows us to run complex methods of analysis like we saw today with a user-friendly interface that hides the complexity of the algorithms behind every single step into a logical, color-coded, and clear path from raw data to structured data, and then gives you also access to tools for annotation and statistical analysis, as well as machine learning to actually conduct your full raw to insights, right? Raw data to insights. And as you can see, we recently added a new section right here called single cell analysis. You can hover over each section and you will see here with, and as expected, we're going to be dealing with UMAP and t-SNE plots. So here we have two sections, right? The first section is a section on pre-processing, what we described in the previous kind of few slides. And then the new sections that we are now adding are advanced downstream analyses, including curated, using bioconductor, and then also spatial single cell analysis. And these sections are going to be introduced during our symposium at the end of this month, March 31st and April 1st. We have a scholar from the Tabor Bioinformatics Research Center who's going to come and present some research projects using some of these tools. But today we'll just do a very brief overview. This platform is included with your Omixlogic training, so you can gain access to this. And then we also have a research license or a commercial license, so you can explore this. But a lot of the methods to learn, you can do this for free. And then we'll show you how to actually run this code. So if you want to do this independently, you can do that as well. So first of all, you have here the option to run different types of data. This is going to be already processed data, right? And we'll talk later about what this data looks like. We're going to skip the upload here, and we're just going to look at this particular section down here. So I'm going to click on start. In the start, essentially your data is going to come in like 3 or 4 different files. So you're going to have your gene matrix, you're going to have your barcodes, you're going to have your metadata, right? So you're going to have all of these different files, and you want to load them in, check them for, you know, whatever data is available so that everything is there. And once you have it, you are then able to process the individual data sets to kind of make sense of them. So the first step is going to be with pre-processing, right? And we're going to do QC. So as you probably see in many of these papers, there's the first step is to really understand how many mitochondrial and how many actual mRNAs do we have, right? That's the first step of QC. And you have here some parameters that you can use to filter out the bad cells, right? You want to filter out the bad information that you have. After that, we talked about normalization, right? So because you have different types of cells, different types of tissues, different maybe quality or amount of mRNA that was captured, you want to then normalize, and you can do that either with log normal data or with SCT. So SCT transformation includes all these 3 steps right here. So log normalize, find variable features, and scale data. Again, here, you can use these parameters as well. After that, we can, if we expect this to be cell cycle dependent, we can use this, or typically we use dimensionality reduction. So PCA, again, number of PCs, we just saw that parameter right there. Run jackstraw, run only on variable features, etc. So we can keep that as is. Then we typically have, again, multiple samples have some change, right? So you have some overlap, but it's not complete. And so to address that, you can use either Harmony, that's without PCA, or curated anchor integration. That means that you identify genes that have similar profiles and you use them as anchors to adjust all of your data. Okay, and here you can use a limited number of features to make it faster. After that, we can perform clustering, and then we can perform dimensionality reduction, UMAP and t-SNE, and compare them, so we can actually select both. And then, and by the way, here you saw that there are some options there as well. Then you want to identify marker genes, right? So again, marker genes is what will tell us, what are these cell types? And we can then either do this ourselves after we build this kind of visualization. We can either manually annotate our cells, or we can use these two methods, this method called CellDex, which can only work for human or mouse data because it works with the database of these human and mouse cell types that are already annotated. Again, you have here a few different types of data repositories that you can select from. Okay, and then optional but possible, you can run differential expression either between clusters or within clusters. So you want to understand, okay, in my cluster of, let's say, immune cells, what are the different processes that I have, or how are my immune cells different from, let's say, epithelial cells, right? So this is an overview of these tools. Again, I'm not going to expand too much on this right now. What I will do is I will pass on. Well, before I do that, are there any other questions that I can answer before we pass it on to Dr. Raghav to talk about how to code all of these independent steps in R? Okay, if you have any questions, feel free to paste them in the chat. I'll be happy to answer them. And again, the T-Bioinfo platform, you can actually check out the section right here. And you can register for free and use the demo pipelines to learn. And you also get an educational subscription together with your Learn subscription for this platform. Okay, so Dr. Raghav, I'll pass it on to you to talk about how to code these methods in R.
Thank you. Thank you very much, Elia. Let me find ways to share my screen and get my video running. I think my video is not being recognized by Zoom. So yeah. Okay, let's, you have to be content with my photograph there. Okay, let's find ways to share my screen. Can you see my screen? Yeah, I think I got my confirmation. Thank you very much. Okay, excellent. Okay. So hello and welcome everybody to the second session of this single cell data analysis, single cell transcriptomics data analysis webinar. And this session is going to focus on the downstream analysis of the single cell data with packages like curated and mostly with packages like curated, and how we can implement them in our programming environment using shareable collab notebook. So let's go over some of the already discussed introductions that we had. So why is there a need for single cell RNA-seq analysis, and how is that different from bulk RNA-seq analysis? So we already found out that single cell transcriptomics profiles or it quantifies mRNAs present in each and every single cell, and this is widely used in the single cell profiling techniques like that will help us identify different cell types that are present in the present in our sample, and also understand the heterogeneity and different categories or subcategories or different population of cells that are present in the sample that we have, based on depending upon the or by analyzing the expression variation between these cells. So unlike bulk cell RNA-seq profiling, the sequencing libraries there in the bulk cell are generated for thousands and thousands of cells, and we have an average gene expression that will that comes out of all the cells. So for some studies, like understanding the difference or general difference between, let's say, tumor cells or expression patterns between tumor cells and normal cells, bulk RNA cell techniques are very helpful and useful and are employed in those in those methodologies. But in order to study different tumor microenvironment, for example, or different cell differentiation patterns or different differentiation patterns for a cell across the cell cycles. So bulk RNA-seq analysis might not be very helpful, and we have to resort to single cell RNA-seq analysis techniques. So as yeah, so this technology helps us to measure the transcriptomic information for a range of thousands or even up to a lot of millions of single cells in one single experiment. And by measuring these transcriptomic information that are specific to different cells, we can reveal the nature of heterogeneity, or we can understand the stochastic expression variability that can be linked with the phenotypic characteristics of the cell that is quantified. So we already discussed major differences between bulk and single cell RNA-seq methods. To highlight few of them, so historically, bulk cell RNA-seq is a major breakthrough that replaced microarray technology, and it has been widely used to measure average expression levels for each gene across large populations or large population of cells in a sample. So these methods are useful for comparative transcriptomics analysis, like as I already mentioned, examples of the same tissues from different species or samples of the same tissues from different spot or different conditions. So this is also helpful or in quantifying gene expression signatures for ensembles, or in disease studies where we will focus on understanding gene expression signatures for the disease across different cells or different cells that exhibit the same disease stage. So, but this method, as we understand, is insufficient for more revealing studies, or to understand the heterogeneity of the system, or to understand complex tissues within, for example, say brain regions. So for these purposes, single cell RNA-seq technologies are very, very helpful, as they measure the distribution of expression levels for each gene across the population of cells. And this technology, as it states, will allow us new biological, or to study novel biological questions in which we can ask cell-specific, or we can investigate cell-specific changes in the transcriptomes to identify different cell types, and to characterize, for example, gene regulatory networks across the different cells, and also to study the stochasticity of the gene expression pattern across different cells. So major applications of single cell RNA-seq technology includes, as I mentioned, studying heterogeneity, and to trace the evolution, or trace the cell differentiation cycle across the time, and also to understand, we already discussed about this, the stochastic nature of the gene expression pattern across different status of the cell that we have. So if we can generally summarize the whole single cell RNA-seq quantification and analysis process in general, so some of the key steps involves, as it has already been discussed, is to extract cells and to isolate these cells from from the tissues with technologies like Droplet and SmartSeq, etc. And then what follows is some of the steps that share their library preparation, amplification, sequencing, and generation of structured count matrix with the bulk cell RNA-seq technologies. So, and then we can perform a downstream analysis to identify for different cell types, or to identify novel biomarkers or novel markers that will characterize different cell types, and also to construct cell trajectories in the differentiation processes, or in the study of differentiation of the cells, etc. So the analysis pipeline, while can be very broadly summarized, as you are already know and gotten introduced to the extensive level of different algorithms involved in this pipeline, but this is a very broad summarization, can be summarized as upstream analysis that will constitute until the generation of the count matrix from the sequence read data. And then what follows thereafter can be termed as a downstream analysis. And some of the major steps that involved in this pipeline will include quality control, which will filter or which will select good quality cells and genes that express high variability, or genes that are expressed in high variance. And these selection or quality control steps are very crucial for the clustering process, and also for biomarker or marker gene marker identification that will characterize the cluster and hence help us in annotating these clusters to different or specific cell types. So further down stream, after the generation of these count metrics, we can use the downstream analysis to, for example, study the trajectory, to do or perform trajectory analysis, and to study trajectories of different cell differentiation. And we can also perform differential gene expression of marker genes across the cell types or across different clusters. And also we can do or perform identify novel marker genes that will characterize these cell types using packages like curated and mainly using packages like curated. So as we already found out, the upstream analysis can be performed in tBioInfo server by setting up a pipeline that is similar to this, where that include pre-processing, mapping, and assigning of the reads, and identifying different cells, and then quantification of the gene expression levels for different cells. So for a typical pipeline like this, which will take in the FASTQ files and then produce structured count matrix data, the output will look like, or the output that is generated will have the count matrix table that can be subjected to further downstream analysis. So if we download this count matrix table and we can open it up, and this is what we see. A typical count matrix will look like this, where we have columns of where the columns of the tables are the cells as represented by the barcodes in their header, and the rows are transcript IDs as represented by their ensemble ID names. And what we have in them is the expression level for these gene expression for these particular transcript across or for specific cells or different cells that we have. And one striking difference that you see, for example, when you compare a count matrix table to general gene expression table of bulk cell RNA-seq analysis, is the number of zeros that we have in the count matrix table. And that this will quantify or qualify, this will qualify as a sparse data or sparse table, so for which we have to include imputation methods and further methods in which we can from which we can make use of this sparse data and perform further analysis. So to do this, or hence since this table is sparse and it includes lots of zeros, and there is a general asymmetry in the expression level of these genes across different cells, then many of the analysis techniques that we are going to use will be non-parametric and non-linear technologies or non-linear algorithms. So so from these we can actually identify, or we can use a database of known molecular markers or known molecular markers that are genes that will associate these genes to specific cell types and can be used to annotate the cells present in the count matrix, or the cells that we observe in the count matrix, and generate a table that looks like this. So from here, with the help of nonlinear dimensionality reduction techniques like TSNE, UMAP, we can visualize the segregation pattern, or we can visualize distinct clusters of cells that are present or that the data in our count matrix describes. So these distinct clusters that we can identify based on the segregation pattern of cells uh, um, as exhibited in the gene expression table, we can identify specific cell types or specific clusters that can be annotated by two cell types based on the marker genes that we have used so far. So this is a very broad step or overview of how downstream analysis can be helpful in and understanding the cell types. Let's go over these steps one by one in detail, and what are the different um, analysis processes that we can subject our count matrix to, and what we can learn from these analysis processes. And then we will go to a practical demonstration of how to perform this in our environment. So for this data, we will make use of the PBMC, or peripheral blood mononuclear cells data set that is freely available from 10x Genomics in this link. So all you need is to sign up, or all you need is to mention your name and associated institutes or associated email address, and then download this data. It is freely available, and it is we can make use, or we can use this for our further analysis or downstream analysis in the Google Colab notebook. So when or while downloading this data, the 10x Genomics actually provides us a summary of various statistics that are that we can observe from this PBMC data, and also it can help us to compare it can help us to compare what are the recommended statistics, and based on that we can identify, or we can immediately notice that the data that we currently have satisfies all the recommended statistics or required recommended statistics for performing a reliable downstream analysis in the with the data that we have. So some of the key potential problems that we have to keep in mind before proceeding further are those general potential problems that are associated with the single cell RNA analysis methods. So, for example, the amplification efficiency, and hence because of that, the quantifiable or detectable gene expression levels are typically very low or less. So this low amplification efficiency will actually place the number of genes or the transcripts per cell very close to a technical noise level, or very close to a level which can be characterized, or which can be defined as technical variations. So we have to find out, or we have to leave out those transcripts which exhibit expression levels on this side of the curve. So we are going to consider only for those transcripts for which we have very high expression variation or amplification efficiency. So we further have other technical issues like gene dropouts. We can have missing cells from the droplets, or we can have ruptured cells, or we can have fused droplets where more than one cell can be present in one droplet, and all these can potentially complicate the analysis pipeline. And hence you will see we have to perform multiple rounds of quality control, or multiple rounds of threshold setting, or identifying threshold, or efficiently filtering problematic data from the downstream analysis to make some sense of these data, or to understand and identify patterns that can be reliably related to the phenotype or biological phenotype that we have for the sample set, for the sample under study. So keeping this in mind, once we have the count matrix, and we can perform this curated based analysis in the Taubot bioinfo platform pipeline also. So some of the major steps has already been gone over by Ilya, include specifically include to mm, quality control, to perform quality control, and to select or to leave out bad quality cells, or to select only good quality cells for each sample, and to normalize the data for each sample, to correct for the batch effects, and to correct for other problematic variations in the data. And we further we have to reduce the dimensions, or not reduce the dimension, we can say select the specific dimensions or number of features based on the very high variability that it can that it can represent, or that it can show. And then we have to perform integration, or we have to use nonlinear techniques like t-SNE and UMAP to visualize the sample segregation, or cell type um, clustering, or cell type separation in the nonlinear dimensions, or in the represented or reduced dimensions. And then to do this, we have to perform proper scaling of the data, and we have to perform centering of the data, because without centering and scaling, we cannot perform PCA or any dimensionality reduction techniques. And then we can further use batch effects, or remove batch effects using harmony methods, or fast MNN methods. And we can select key principal components, and then by selecting key principal components, we can also identify those specific genes which contribute most to the significant principal components that will be employed in nonlinear dimensionality detection techniques like t-SNE and UMAP. And then what follows is, after this visualization, we have to perform clustering. And in clustering of cells, we will identify and find different clusters. And the goal in clustering, as you had already been introduced to, is to identify, or is to place similar cells together in a very low dimensional space. What happened suddenly? Okay, yeah. So, and then we can, after identifying or after characterizing the sample segregation based on different clusters that we can visualize, or that we can identify in our map, then we have to assign these clusters to cell types, or annotate these clusters to different cell types based on the marker genes that are different, or that qualify for, as significant genes that can differentiate between different clusters. And then once we have performed this annotation, we can further parallelly perform a differential gene expression, or differentially expressed genes, identification of differentially expressed genes between clusters or between cell types in our downstream analysis. So all these methods we can perform in a pipeline, or also we can perform in using R's environment by installing a package, or installing one package called curated, which has the ability to perform all of these analysis in the using very highly sophisticated visualization techniques. We will go over that, and let's see. Yeah, so one of the important steps, as it has been discussed multiple times, is to remove low quality samples, or is to mention a threshold based on which the low quality samples are removed. So how is it done? So it is usually done by understanding, or by plotting the number of genes across different samples, and then this can help us in understanding cell doublets or cell multiplets, which will exhibit an aberrantly high gene count. So most of the gene counts here for these samples are centered around this 1000 to 2000 range. So whenever we have samples that have really very high number of genes, then we can use a threshold based on which, for example, let's say if I use a threshold of 2000, then that 2000 threshold we can identify from these quality control violin plots. Visualization. So this will be different for different data. So for this data, we can use a cutoff of, like, say 2000. which will cover most of the cells, most of the samples, and most of the genes, above which we may suspect that we have different problems, or we have low quality cells that we do not want to consider for our analysis. So the percentage of mitochondrial genes is also very good here. It is on the very lower side. So what will happen is whenever we have cells that are ruptured and their mRNA molecules that escape from the cells from the cell, then we will have the only available genes are from the mitochondrial counterparts or mitochondria. So for those cells, we will have very high percentage of mitochondrial genes, and hence we can understand, or we can identify, or we can we can find a threshold based on which we will want to filter out the percentage of mitochondrial genes for which we suspect the cells may have been ruptured, or the cells may have been dead or of low quality. But this also you have to keep in mind that this is not the case for most of the this is not the case for all the biological situation, where for example, when you have yeah for example, sorry, can you mute yourself? Yes. Hello. Yeah. So for example, when you have for example, specific biological systems, where when you have when you study cardiomyocytes or different cell types, where those cell types generally exhibit a very high level of mitochondrial reads, or very high percentage of mitochondrial reads, then we have to understand the biology of our samples, and then take this or identify the threshold based on which the mitochondrial genes are being filtered out. And then, as we already had discussed in this graph, we have to identify only those genes that exhibit most variation, because when you standardize the variation in the genes across average expressions, then we should not be left with the genes that have very least amount of variation across the cell types, and that will potentially impair our identification, or impair our cluster or quantification of cluster, or impair our biomarker or marker genes identification across different clusters or different gene types. So for this, we will have to ensure that above a standard variance level, above which only above which the genes that exhibit these variance level across the different cell types are considered for further downstream analysis. This is this will reduce a large number of features or cells that features or genes that we have. For example, in this case, in this example, we can see that we are going to filter out something around 4,000 genes, and we are going to use only genes that exhibit most variance, or exhibit most variance in the count, and that count will be around 2,000. So in other way, when we plot this standard variance plot, then we are going to use only top 2,000 most variant genes for our further downstream analysis, that is going to help us quantify, or that's going to help us characterize these clusters, and yeah, that's going to help us characterize these clusters and annotate these samples. So yeah, so where are we? Can you see my screen? I cannot see my screen. Yes, now it's up there. Okay. So once we have filtered out noisy features, or once we have filtered out genes, then we can continue to visualize the presence of global structure or global pattern in our data by performing clustering, and to study variability across these clusters or across the different cell types. So we perform clustering mainly to determine, for example, a subset of genes that can characterize different cell types, or we can determine the variability and difference in the expression pattern across different cell types based on our data. So this clustering will be very efficient, and all these processes will be efficient only when we have filtered out a lot of noise from our data, and we have subsetted, or we have selected only very few top quality genes or features or top quality cells to focus our downstream analysis to. So to understand the number of informative clusters we might that we might find in our data, we can make use of something called as elbow plot, where the variance expressed or explained by components, and the number of clusters can be plotted, and then the goal is to identify the change in the curve pattern or change in the curve, where after this specific point there is no meaningful separation can be achieved by dividing the available data or available distribution into different clusters. So based on this, we can make a decision that yeah, maybe this is around 10 or 11, I think, yeah, and we can use 11 as a cutoff or 11 as a goal to identify different clusters in our cluster map or in our dimensional reduction techniques maps. So so we already discussed a lot about nonlinear dimensionality reduction techniques like t-SNE. So this is a clustering result of t-SNE plot visualization for the data, for the for the data that we have, and the number of clusters that we identify comes from this elbow plot. So we want to identify 11 different clusters. That might not be all the 11 clusters might not be relevant in this case. So this could be an iterative process. So here we want to identify 11 different clusters, and then the data is good. So we can actually see the presence of most of these clusters as separate entity and not as not as distinct or not as scattered points. So in addition to t-SNE, we are also got introduced to UMAP and other nonlinear based dimensional reduction techniques, which will in essence use the top principal components to visualize these patterns, right? So that is what we will use to speed up this process of getting or identifying these clusters and using a nonlinear based or nonlinear differential technique. So let me quickly yeah, yes. So what do we have next? Then next, what do we have is to understand the gene expression or different marker gene expression pattern that are specific to each cluster. So this can help us annotate the clusters based on the cell types, and further we can also understand, or we can also perform a differential gene expression analysis for the marker genes across different clusters or across different cell types. So this is a very very poor and broad graph, and I will show you another graph which will take only a couple of examples that will be very clear. So more useful representation like dot plots can be helpful, can also be helpful in understanding marker genes that are specific to different clusters or identity here, and when you have a larger data sets where we have a lot of cells, and box plot kind of or violent plot kind of representation can confuse or can be insufficient in identifying or in comparing the patterns of these marker genes across different clusters. So for those cases, it is recommended to perform dot plots where the plot naturally shields the noise that are caused by very low expressed genes or dropout genes. So when you have a lot of cells, for example, a lot of different cells in this case, a dot plot is more informative and useful than performing or than visualizing the gene expression pattern across different cell types. So this, we have only 11 clusters, so we have only 11, but when you have a lot of different clusters, then the complexity keeps on increasing. So this is annotating based on the marker genes that we have identified. So first we have to identify different clusters, and then we have to identify the marker genes that characterizes these clusters and are not common to other clusters. And then based on these marker genes that will be that are specific to specific clusters, we can identify the cell types that are identify the cell types and then annotate these clusters with the cell types. So this can be performed. This can only be performed after identifying the marker genes. And once we have identified the marker genes, we can use the annotation and label our clusters based on the annotation that we have identified, and that will be helpful in understanding the segregations or segregation of these different cell types in our based on the gene expression pattern that we have. So yeah, we can also use uh, heat map based method, which is for which uh, the differential expression of these markers across different cell types are actually calculated, and the differential expression analysis is very similar to the method that uses HR for bulk RNA-seq analysis. So when we use this heat map um, mode to visualize the most differentially expressed genes, we can potentially identify novel biomarker genes or novel marker genes in addition to the already existing marker genes like here, or we can also observe patterns for identifying additional cell types based on the gene expression data or pattern that we have here. For example, this does not, this is a very high quality data set, and this doesn't give out any additional cell type or cell patterns. But yeah, when you see a marker difference between or within one cluster, then those are considered as hints for the presence of more than one type of cells or more than one type of more than one type of cells in that specific cluster itself. So yeah, as I mentioned, if I zoom into or if I select only a couple of genes for the violin plot representation, then we can easily identify the expression patterns that are high for these for which the expression patterns for these specific genes for different clusters, and then we can say that okay, for cluster 10 CCL4 is a marker gene, and similarly for cluster 11 this gene is a marker gene. So so when we have a similar expression pattern for the genes across different clusters, so obviously they cannot be employed as marker genes, and so we can use from this information, we can use some additional analysis of marker genes identification for different clusters. Then so this is, so far we have been dealing or discussing with samples or single cell RNA quantification methods that is measured for sample for one condition. So we can also have samples that are for which single cell data is recorded for multiple different conditions. So when we have such sample, or we can have a single cell RNA data measured for samples for multiple different time points, then we have then we can perform perform a differential gene expression analysis across different conditions, and then identify, or we can trace the level of expression across these different conditions, and then relate that to the sample state that we have. So yes, so this is also very very useful visualization where for example, in addition to what we see here, when we use only selective marker genes, and then visualize the presence of those marker genes to characterize or being characteristics of different clusters, then we can say that we can see their pattern or their expression pattern as observed in different clusters. So this is also very useful mode of visualization for the t-SNE clusters, or for the UMAP different non-linear dimensional t-reduction technique clusters and identification of marker genes. So we will do all this, we will perform all this in the Google Colab notebook. So I can actually stop here to ask for some questions if you have if you have for the session until now, before going to a demo mode. Please let me know if you have any questions or if any queries, and then I can proceed to the demo mode. Do you have any questions? Or is everything clear? Maybe with the demonstration, you will have much more freedom and have a lot more clarity. Shall we proceed? I think we can, yeah. Yeah, okay. I think we can proceed with the demonstration. I will stop share and share my Google Colab notebook. Okay. Share my screen. Yeah, please confirm that you can see my screen or view my screen.
Yes, sir, we can see your
Okay, yeah,
screen.
perfect. Thank you very much. Yes, so we are going to do everything, and also we can do much more in the curated libraries and packages in the using this Colab, or also you can install the same in your R Studio, which is recommended because this can take a lot of time to perform. So you have to, if you don't have the curated package, you have to install the curated package, and this is optional. You don't have to install the curated data, but yeah, other packages will include ggplot2 for visualization and patchwork for other variance identification, dplyr for general ease of visualizing the table and other data. So let me quickly ask, how many of you have decent experience with R and R Studio? Recent programming experience with R and R Studio. Please, yeah, please, yes, or include something in the chat window so that I can go through. Yes, excellent. So that will help a lot, because yeah, so if you do not have a lot of experience in using R as an analysis platform for your downstream analysis, then we have a lot of introductory courses in R, which will help you in a step-by-step manner how to load data, for example, and how to use different libraries to visualize, etc., etc. That will be very handy here. But for this specific analysis, we are going to limit ourselves to loading curated and other packages, and most of the functions and everything is very well demonstrated or very well developed in curated package, and they have an excellent help and they have an excellent help site which can be used, or which can be utilized for further knowledge. So let's go over this, and let's load up the libraries. Just loading the library. And for the data that we have, this data is the, as I mentioned, PBMC 3K data, and where they have around 2,700 single cells and 13,000 or 14,000 genes per cells, or not per cells, for these different features. So if you go to this specific link, so that link is going to okay, since I have already logged in, this is actually giving me to help asking me to download this. But usually when you click this link, or when you go to this link, it'll ask you to fill out what's your name, what's your email address, etc. After which you can download the data. And then after downloading this data, you can unzip, and then we will find, let me share everything. Yeah, and then yes, yeah, I think you can see all my screen. So after unloading this, then you will generally find these three files. Can you see my screen? I think you can see my screen. Yeah. So the matrix.mtx is the count matrix, or the output of the count matrix that you will generate when you perform the upstream analysis. And barcodes are the barcodes to identify different cell types, and genes are the different genes that are associated with the ensemble ID that we have. This will be very helpful. I will tell you how in the downstream analysis. So these three are the files, and the barcode and matrix files are absolutely necessary for performing downstream analysis. If you don't have genes, then we might be hindered in identifying the genes associated with ensemble IDs, but it's not very difficult to relate, or it's not very difficult to get a list of genes that are associated with ensemble IDs that we have. So it's easy to make these. So these are the 3 files that you want to upload. Where do you want to upload in this collab when you get hold of this collab? So you want to go here and then open up the folder. And if you see a sample or any other content or any other folder location, you have to go to the root folder. That can be, for example, let's say if I'm inside this, how to open this. Okay. Yeah. If I'm inside this, then I can press dot back and then go back to this root mode. And then I see here, I have uploaded these files directly here using this specific upload to session storage option. So this is where you will tell the code to find the input data, which will be absolutely necessary for our downstream analysis. Let's go. And then this is where I'm telling the data where to find the read 10x data, where to find the read. I'm using. Can you please mute? Yeah. Can you please? Yes. Thank you very much. So we have to create something like a curated object, which will take these count metrics and then take this barcode and take these genes and then combine everything together, and then it will create a data frame like object that can be employed for further curated based analysis. So let's run this. This is very simple, and when you use this data for for visualization, this is what it looks like. Let's open up a new one. We can say, I hope this will still work. Yes. So if you see the dimension of this curated object or data frame that we have, so it has 13,714 features or 13,700 genes across 2,700 cells. That we have here. So we can, for example, display a few genes and then see what are their expression patterns here, or we can also use head. And then see, that is what you have here. You have a data frame of number of counts we have, number of features in this one. But if we use specific genes, then we can identify or we can see their expression pattern across the different cell types. And here is the example of the sparseness of the matrix that we observe or that we see. For example, this is in the counts. If you go to, instead of head, if you use, let's say 10 to 3, okay, 0 variable features. Why is that so? Because it's the other way around. Okay, I think I think I have to do some dot data. Yes. So this is what I wanted you to see. So you have a lot of sparseness in these in this data. Let's see. Yeah. So generally, you have a lot of sparseness in this data where you have expression patterns or expression levels only populating a few of the cell types, and not all the cell types are with the data. So yeah, we can use different heads and different display options to peek at the data. And then we have to quickly go to quality control metrics. One of the quality control metrics that I mentioned is to identify the mitochondrial genes or mitochondrial gene percentage genes, and then get rid of those mitochondrial genes. So how we can do that? We can actually use the grep command, or we can use the pattern with which all the mitochondrial genes are actually encoded, or have their gene names that starts with uppercase M and uppercase T hyphen. We can use this pattern to match for their presence in the feature set, and then we can get rid of those mitochondrial genes, and that's it. Yeah. So when we that can be done, and first we and then we can take these match these pattern, and then create another column that is going to count, or that is going to quantify the amount of mitochondrial DNA or mitochondrial DNA percentage present in each and every cell type. So this is the input file that we will use for our violin plot, which will tell you whether the percentage of mitochondrial data, mitochondrial gene, or the percentage of different DNA for the samples, or unique UMIs for the samples, how many different ones that we have, and then we can set our threshold based on this. So so when we plot the violin plot, we will know what we want to plot here. Then we can use this to identify it. So I encourage you to go to that website, or I can also give you a link of another data set, so you can perform the same analysis for a different data set. So this is a difference to show you how, for example, normal genes and their expression patterns, expression level, or count level vary between normal genes and also between mitochondrial genes. So when we have you can simply plot a scatter plot between number of counts of the gene counts versus percentage of the mitochondrial DNA, and then we can make a scatter plot between the expression counts and the number of features that we have. And this can also help us tell that the percentage of mitochondrial genes present in our data. So this will also help us visualize the relationship between the amount of mitochondrial genes present in our data. So for example, here, so this is where we are going to employ the filtering, or this is going to apply the quality control filtering that we will identify from these box plots, from these box plots. So we can say we want to have the number of features of RNA should be greater than 200, and it should be lesser than 2500, and our percentage of mitochondrial DNA should be less than 5%. So we don't want a lot of zeros in here, so we want to have 200 to 2500, and that's a block that we are employing for from this violin plot exploratory analysis. So this is where we are rewriting our data set by subsetting the data set of this, and let's see if I do the dimension, how much have we reduced. So we have not reduced a lot. When we go up, I think, okay, from 2700 samples, we have filtered out something around 400 samples, or only 78 samples. So we have a very high quality sample, and that we also can visualize from this data, where we don't have a lot of dots above the 2500 and in the upper region of these plots, which is representative, or which is what you will observe for data that is not very good. So after this, so we have, what is it? So we have performed this filtering, and then after this filtering, we have to perform something like normalization, and so that we can use visualization technique and dimensionality reduction methods to our data, and we also can compare expression measurements across different samples and in a normalized scale. So we perform normalization, and we put a scale factor of 1000 or 10,000, sorry, and then we will actually use this variance stabilization technique, which is the same method used in something like HR and DSEQ2 analysis, to understand most variable features. This is what we spoke about in the in identifying most variable genes. So this actually tells us, if you can see here, you can see that it's 13,000 features that we have, and after performing this, and yeah, after performing this, let's see. See 3. Yes. So okay. Yeah. So we have only, okay, there's something is missing. Okay. Yeah, we are subsetting this to a number of features that is greater than, yeah, that is for greater than 2000 variable count. Yeah, so non-variable count is 30. Yeah, something is missing, fishy here. Let me come back to this later and I address this, what happens here. So so after this, we will use this variable plot to plot which are the genes that we are, which are the variables or variable genes that we are actually counting to further analysis. So this is some of the top mm, genes with their annotation present here, and we can now perform scaling of the data. We have normalized the data and we have identified top variable genes in the data. Then we can perform a scaling of the data and then do a PCA on the data to identify top genes that are going to contribute to defining the different principal components for our data. So let's scale, yeah, let's use scale data function to scale it, and then we will run PCA to do it. A lot of these functions are slightly different from what you are going to see. Yeah. I will share the notebook. Yes, I will share the notebook. Yes. Let me. Okay, this is the notebook that you can go through, make a copy of this, and yeah, and go through. Sorry, let me. Yeah. So after performing PCA, then we have to decide upon number of components that we want to use for our nonlinear dimensionality reduction methods and techniques. So for that, we will quickly, for example, print out different genes that are very that are specific or that are characterized, that characterizes the principal components P1 and P2. So these are called dimension loadings, and if you see the loading plot in normal PCA, where you will have different um, features that are that will characterize different principal components and how they are closely related to each other. So this is a different this is one another visualization pattern for the different dimension loadings. Then we have, for these two dimensions, we have, these are the genes that are that contribute most to characterization of the PC1 and PC2. So finally, we plot the we plot our data using the principal components that we have calculated, and even with the principal components that we can see, our data exhibits some sort of structure that is very broadly, maybe I can say 4 clusters identified, but this is where we stop with the linear dimensionality reduction technique or representation like PCA. You can make even view axis plot. We can make a wait, let me, yeah, I think you can make a copy of this. Let's see. Okay, get link, copy link. Yeah, restricted to only open with, okay, no, not like that. Anyone with the link, yeah, viewer, done. Okay, anyone with the link, you can open this collab. Yeah. So so we do, basically we don't have very many different clusters, and we don't have a method to identify clusters from PCA. So for that, we have to resort to non-dimensionality reduction techniques like t-SNE and UMAP. And based on PCA, maybe we can do something like dimensional different heat maps for the for these PC according to the dimension scores, and this will actually help us decide upon the number of principal components that will that we can employ for our further nonlinear dimensionality reduction techniques, because that uses the top 15 or top 20 PC principal components that we are going to mention, or that we have the option of mentioning. So here, if you see that the heat map after something around, let's say PC8 of 8 components actually fizzes out, and there is not much identifiable pattern left. So we can reduce the number of principal components to be used and analyzed to to 8 for the UMAP on this one. So this is, as we already saw, Jackstraw is a very useful method to identify the principal components that will, for example, that will qualify based on their p-value to differentiate between the different cell types, or differentiate between to be contributing to the variance that is observed in the original data. So this is a Jackstraw plot, but mind you, this is going to take long time to compute. So that's why I've already computed. So if you start this will, for example, in my computer for this connection, it took 2 minutes, but it can very well take, I've had sessions where it took something like 10 minutes to calculate. So for this, for the Jackstraw plot, you see, you observe this cutoff value here, and that determines the different principal components that we can employ essentially for further dimensionality reduction techniques or t-SNE and UMAP techniques. So we see that if this 10 to 12 PCA is not very visible here, or we can see it here, and the normal p-value cutoff 0.05 can be employed here, which will qualify first 13 principal components, or 0.005 can be employed, or even higher cutoff can be employed, and we can see that first 12 components has a very high or very low p-value, very good p-value score. So we base our selection based on this observation here. Also, we can do the same using the elbow plot. The elbow plot as I can show you the deviation, then one might standard deviation, then one might relate to the number of principal components that we have. And this tells that we could have actually had good results by stopping at 8 or 9 principal components. And then to enable clusters, then we have to find neighbors, and for this we can employ for example UMAP, and before that we can find clusters based on the scores, or based on the Jackstraw scores that we have computed from our PC map, principal component map. And from these scores, first we have to identify the nearest neighbors, and there are multiple different ways of clustering, and this is one of the best way. Identifying nearest neighbor method is the best way to perform clustering for example, say nonlinear dimension methods, where they also cluster, or they also display segregate samples based on their neighborhood, or based on the nearest neighbor calculations. So we find the different clusters, and then we can look at the different clusters, or identity, or idents would mean clusters. So this is first 5 cells and their cluster level identity. So this is cell 1 with their identity 1. These are the cluster 1. There's not much difference here, but we can when we go down, we can see. So we can perform UMAP. We can run UMAP, and then we can try to identify different clusters we have. So we have been looking for different clusters, and then we can see that straight away see the difference between linear transformation technique like PCA and non-linear transformation technique like UMAP. That will work much better for the data that we have. So this we don't need to do this, and when we go down, we have to use the UMAP identifier distribution to find clusters, and we can use this to identify marker genes that are for clusters. So find markers function will do that. For that, we have to give UMAP object, or we can give PCA object, or we can give KSNI object. So much more preferred methods or preferred objects are UMAP and KSNI for their ability to distinguish between the our ability to display the structures that are hidden in the data set. And yeah, this will tell for which cluster we are going to find marker, and this is going to tell a minimum percentage of the genes that has to be found in the cells that we have. So 0.25 would say that if I identify, let's say, these genes as to be marker genes for my clusters, they have to be present in 25% of the cells that contribute to these clusters. So we can do that, and we can identify these are the marker genes that is present in the in these clusters, and then we can perform the same for next cluster or one more cluster this way. But we cannot obviously do this for all the different clusters, so we can use find all markers and use similar threshold like minimum percentage of identifying or minimum percentage of observing the genes in the number of cells that contribute to the clusters. And also we can use fold change threshold also in this, and then we can visualize their clusters by grouping by clusters. So and their fold change, and then we have for example 8 different clusters and their marker genes that that will characterize these 8 different clusters. So since we have for example maximum number of marker genes that we have used only 2, so we have displayed top 2 marker genes for each cluster. So that's what we have. You can change this number to 3 to display top 3 genes for these clusters, and their average p-values and average percentage contribution and average log FC log fold change can be identified here. So and then yeah, we can actually visualize these markers. As I mentioned, we can actually visualize, for example, these features or markers, selected markers for different clusters. This is based on logarithmic scale expression level. We can do the same for counts, raw counts also. And then this is one of the most interesting feature plot that we will be used to. We can employ to identify or characterize these features or these clusters and their ability to annotate or to identify different clusters. So a feature plot will do this, and then we can use the heat map visualization method simply to see how different features are actually contributing to visualizing the difference in the expression pattern that is observed by different clusters. So now we have to assign cluster types for different clusters. So this has to be done by, for example, this has to be done externally. So we have to take, for example, these marker genes and identify the cells that are that these marker genes are characteristic to, and then we can take those cell types and then assign it to the cluster ID, and then we can use the new cluster IDs or the names of these identified cell types to visualize their presence or visualize their segregation in the TSNI or UMAP visualization pattern. And yeah, and this is this we can do. So this one we have to do it externally. And there are, I think there are also other databases available to do this, or if you give the marker genes that will actually identify different cell types also that are also available. So I can take some questions when you have, because we have already exceeded a lot of time in in explaining this. That's why I did not run everything, each and every step. UMAP and other techniques will take a long time to finish up in this code block. Yeah. So let me take few questions if you have, and I will let you go through these code block, and we can actually come back the day after tomorrow. And if you have any queries, you can discuss those queries, or you can contact me at support@find.bio website. Let's see.
Thank you so much, Dr. Raghav, for such a wonderful session. And it was good to
Sorry,
see
sorry, Sonalika. Sorry to interrupt. I will actually include a link for another data set in the Colab notebook, so you can have the freedom of trying this out in a new data set. This is also a 10x based data set. I think it's a it's a brain different cell type data set. So you can visualize how different data set can be employed in a Surat package to understand and annotate different cell types. Now, yeah, over to you, Sunilika. Sorry, I forgot to tell this.
Thank you so much, sir. And it's good to see the participants still like being here for 3 hours and going through this extensive workshop. I hope you were able to absorb the concepts and go through some of these things. And we'll share the recording with you, definitely. Go through the concepts once again, revisit all the things that we discussed today, practice them, and then when you have any questions, get back to us on the mail ID that Dr. Raghav has shared in the chat box. It's support@pine.bio. You can also reach out to us at marketing@omixlogic.com. Okay, so now I wanted to discuss about the registration steps. So the first day of the workshop was definitely a free event for everyone, those who have an interest in single cell transcriptomics and also transcriptomic data analysis. So we wanted to have you all join this workshop and get an idea of what all the workshop is about and how you can work on these concepts and also get some hands-on, right? So today was just an introductory session and it's just the beginning. We have the other 2 days for the workshop as well, where we will dig more deep into these concepts and get a better hands-on analysis, an extensive one, and get working. Also, the people who register themselves for the workshop will get an access to the resources, as Ilya was telling, that you will have the access to the Omixlogic Learn courses, all of the courses, and also the educational license will be given on the tbioinfo server, where you can upload your own data from the big data repositories, the single cell data, and also the transcriptomic data definitely, and work on it. So now what are the steps to get yourself registered for the workshop? Now this is a 3 days workshop, but the access to the portal, the Omixlogic Learn Training portal, the R lessons, and also the tbioinfo server will be for a month. So this is one thing that I wanted to make it clear. Now let me just quickly put the link to the program page in the chat box. So this is the program page where you'll be able to learn more about this particular workshop, and also you have the whole schedule, right? So today we had 2 sessions. Tomorrow we have the session on what is single cell RNASeq, and then we have a session. We have 2 sessions on 10th March as well. I think one of them is missing from here, and I'll add that up here. Also, I'll share the whole schedule of the workshop once again via mail. Now, if you scroll below on the same registration page, you'll find 3 subscription levels. One is a beginner one, the other is intermediate, and then we have an advanced level of subscription. So now what are these subscription types all about? In the beginner one, you will have the access to the sessions that we already had, like the ones today. And in the intermediate subscription, you will have the access to the symposium. You will be able to become a part of the symposium that we have on March 31st and also April 1st, where we'll be having various speakers from various organizations and also international institutions and universities. And they'll be speaking about these research trends, data science, bioinformatics, the role of omics data analysis, and also you will be able to become a part of the poster presentation competition if you would like to, and present your research work. It has to be your own research work that you can definitely present in the poster presentation competition. So taking the intermediate subscription level will help you become a part of this research symposium that I just told you about. Now, to learn about the symposium, who all will be the speakers, who all would be speaking, what all you will be able to learn, and what it is all about, you can visit the page here. And putting it in the chat box also, I'll share via email. And then you have the advanced level. And in the advanced level, you will have the access to one-on-one mentor sessions. So these are group sessions that we have as of now. And in the advanced subscription, you will be able to meet the mentors one-on-one and take your questions and align your research interest or any research proposal that you might have and get started with your own research work. So that was about these three different types of subscription levels that you can pick for yourself and become a part of the workshop and leverage the resources that we have on the portal and get the access to the educational license of the server as well. Now, talking about certification, yes, definitely this workshop is certified, and after completing the workshop, you will be able to download your certificate from the program page itself. So day after tomorrow, when we have the third day of the workshop and we'll be completing the like all the resources and the sessions, I'll tell you like what steps you need to follow to get yourself certified for the workshop. So that was all that I wanted to discuss with you all about getting yourself registered. Only the registered participants will be getting the invitations for tomorrow and day after tomorrow of the workshop. And yeah, so that's pretty much that I wanted to discuss. Now, if any questions are there, you might please put them in the chat box so that we can answer. Okay, so there are no more questions. I think we are good to go and end today's session, and let's meet tomorrow. I am putting in the mail address once again, and you can reach out to us anytime. And also, please make sure that these mail IDs are added to your address book or contact list in your Gmail, or because when we mail out the interested participants, sometimes the mails land in promotion, like in the promotional box, because we have been interacting with a lot of students, researchers, and faculties. So just to make sure that you get the mail, please add these mail IDs in your address book or contact list in Gmail. Okay. Thank you everyone for joining, and thank you so much, Dr. Raghav, once again for such a wonderful session. It was nice to have you all with us here today, and looking forward to our next session tomorrow. Thank you everyone. Have a great day. Thank you, Ryan. Thank you, Sri Gauri.